Today, we will be looking at a process known as translation, and more specifically, how the correct amino acids are brought to the ribosome by tRNA in order to produce a protein. We will also look into how just one little problem with this process could result in negative consequences for our cells. When most mRNA is produced in the nucleus, it is then shuttled to the cytoplasm of the cell. Here's where tons of ribosomes live and work. Oh, I'm getting a call. The mRNA just entered the cytoplasm. Let's go get our ribosomes ready.
The specificity of each tRNA molecule for each codon of the mRNA molecule was deemed the adapter hypothesis by Francis Crick. But before this realization that there was tRNA, many scientists thought that amino acids could directly bind to the mRNA sequence and form a polypeptide. However, Francis Crick denied that possibility and later noticed that there was an adapter molecule that was capable of binding to both amino acids and to the mRNA. This molecule became known as tRNA. tRNA can be charged or uncharged. An uncharged tRNA means that there is no amino acid conjugated to the end opposite of the anticodons on the tRNA molecule. In order for a tRNA molecule to be charged, the enzyme aminoacyl synthetase binds specific amino acids to specific tRNA molecules. Ribosomes are composed of two subunits. First, the small ribosomal subunit binds to the beginning of the 5' end of the mRNA sequence, and then the large ribosomal subunit binds. After this, tRNA molecules begin bringing amino acids that will help form the polypeptide end product. Once the tRNA molecule is charged, it can then enter the A site of the ribosome. However, not just any tRNA molecule can bind with any sequence of mRNA. Each reading frame must match with a particular tRNA anticodon carrying an amino acid. After this, the ribosome shifts and the first tRNA molecule is moved to the P side of the ribosome. Next, another charged tRNA molecule with appropriate anticodons can bind to the mRNA and enter the A site of the ribosome. Next, the ribosome synthesizes a polypeptide bond between the two amino acids that the two adjacent tRNA molecules are carrying. After this, the ribosome shifts towards the 3' end of the mRNA and brings in another charged tRNA molecule to the A site, while the previous tRNA molecule shifts to the P and E sites. The tRNA that enters the E site is now uncharged and can leave the ribosome. The amino acid attached to the tRNA that is in the P site can now be conjugated to the amino acid of the tRNA in the A site. And then the process continues until a stop codon is reached. The ribosomal subunits dissociate and the polypeptide chain is freed from the mRNA ribosomal complex. Although there is specificity of which codon of mRNA codes for a particular tRNA molecule and subsequent amino acid, it also comes with challenges when mutations arise. Suppose there was a mutation in the gene that encodes for aminoacyl synthetase. The dysfunctionality could cause amino acids to be paired to the tRNA molecules that pair with mRNA codons that do not follow the universal genetic code. Here, the incorrect amino acid would be conjugated to the tRNA molecule. Although it only impacts one portion of the primary sequence of amino acids here, it could have catastrophic impacts on protein function.